All right, the recording start now. So while people are coming in, we are very pleased to welcome Professor Catalin Voiniciuc to be here with us today. I'm gonna do an introductory slide in a bit, but for those who are already in, so some of you may heard of us from the plant SynBio class that is going on right now. A lot of you may have seen like the advertisement from somewhere else. So like all of you are welcome. We're gonna spend around like one hour roughly through this webinar. And if you have any question, please feel free to type into the chat. Or if you have like further questions, please feel free to unmute yourself at the end of the talk so that we can have some discussion. We plan to have some discussion time together as well. So just to make sure that we are aware of time, I'll go ahead and introduce Professor Catalin Voiniciuc. All right, I'll share my screen real quick. So it's our pleasure to keep going on to our monthly webinar. We had some monthly webinars going on in last year, and then we sort of like on pause because we have to work on the other projects. But here we are back again. Like last month, we have Sebastian Kochoba, who has been working on a lot of the DIY biology, including the plants engineering. So we consider this whole webinar series that we're trying to work on to be part of our plant SynBio work that we are giving up as a course as well. So just a brief information about the course, like we got the micro grant from the Experiment Foundation to work on this course. There are some other infos on this course. I would just keep it like that. If you are interested, please visit the website. There will be some more information. Some of you who are already with us in the plant course, you might be aware of that already. We still have some kind of plan to work on the identifying the plant for whole genome sequencing, and we would do the actual work to figure out the plant genome sequencing in Africa as well. So today we are very honored to have Professor Catalin Voiniciuc from University of Florida with us today. So I met Catalin in person through EBRC conference, and a lot of us were volunteering with EBRC as well. So we had a very great discussion so far. So I reached back to Catalin. We are doing the plant course. So I think it might be great if Catalin can stop by and share his expertise on the plant SynBio with us. And Catalin is very enthusiastic and really looking forward to meet all of you. So with that, I think I'll give the floor to Catalin, and the talk today is going to be on the plant SynBio automation and also on the cell wall issue. So please take it away, Catalin.
All right. Thank you, Ice and Kato, for organizing this wonderful course and webinar series. It's a pleasure to join remotely from Florida. I am involved in teaching synthetic biology to plant scientists at our university, as well as doing outreach for the community. So it is really one of the big advantages to actually reach a global audience. And I hope some of the background that I'm going to share with you, as well as some unpublished data, will be helpful in shaping your future vision about what synthetic biology can accomplish. All right, so my lab is focused on carbohydrates. We call ourselves the designer glycans, and that's because historically I came from the plant genetics side without a formal engineering training. And I was really interested in discovering what are the components of the plant cell wall, what is essentially the most abundant source of organic polymers on our planet, what are their functions, how can we tailor their structure for improved applications. And about 7 years ago, I made a pivot from trying to only use plants to understand the roles of these components to actually introducing some synthetic biology approaches where we use orthogonal or surrogate hosts, such as yeast, to design complex materials like plant carbohydrates from the bottom up. And these approaches can be synergistic, working with plants as well as microbes to address fundamental questions about the biosynthesis and modification of complex materials, would ultimately enable us to enhance crop traits, for example, to have more dietary fibers that have important roles in treating lifestyle diseases such as type 2 diabetes, or to have animal feed that is of superior quality for improving the gut microbiome. There's also important medical applications as well as energy or bioproducts that require custom cell walls. So I outlined some of the applications of the cell walls in this opinion piece from 2 years ago, where essentially you can think of the building blocks of the cell wall as fairly simple monosaccharides like glucose, mannose, which through the sequential action of a series of enzymes like glycosyltransferases, they can be assembled into extracellular polymers that could determine, for example, if a particular tissue is resistant to biotic or abiotic stress, or if that particular wood or other product you can harvest from a plant is ready to convert into some industrial application. The challenge is that all of these applications are time or energy intensive today, and cell wall polysaccharides themselves are challenging to study. In nature, we have a remarkable variety of plant polymers, and within a single species, you may have more than 40 different cell types, and within each cell types, you could have up to 10 or more classes of cell wall polymers. They are difficult to extract. So if you think about, a fallen tree in the forest, yes, you could burn the wood, but it is much more difficult to say, I'm going to make a biodegradable package or extract some nutritional compounds out of that recalcitrant tissue. And then the biosynthesis of each class of polymers turns out to be quite complex. And that's why during my graduate studies at the University of British Columbia in Canada, I focused on a simple model system, the Arabidopsis plant, to try to understand how these molecules are made. And in Arabidopsis, and actually most plant species, about 15% of the genome is predicted to be involved in cell wall biosynthesis or modification. I mean, that is a huge investment of genetic material, and yet relatively few of these genes have characterized functions. That means all those predictions are bioinformatic. It's sort of guilt by association. Now, there's a very nice system in Arabidopsis, which are the seeds. They release a gelatinous layer of polysaccharides known as mucilage, which is produced by a single cell type at a well-defined developmental time point. And my PhD, which was completed in Germany after a master's in Canada, was focused on understanding what are the components of this simplified cell wall, because it is easy to visualize and extract. And it has the advantage that it is genetically dispensable under laboratory conditions. That means you can do the most terrible things to the Arabidopsis seed coat, and at least in the lab, most of those mutants can still germinate and give rise to the next progeny. Now, I'm not sure how many of you have heard of mucilage before, but these gelatinous fibers have diverse roles in nature, and they're found to be produced by all sorts of organs, from roots to seeds to stems and even leaves. And so here are some examples of the biodiversity of seed mucilage. I'm not going to point out all the species outlined here, but they include plantago seeds, flax seeds, chia seeds. Some of you may have had this with your breakfast. That's the example of chia seeds up top. Okra also produces a lot of mucilage. If you slice the pods, you will have noticed the slimy substance rich in polysaccharides. And then some specialized plants like the sundew secrete mucilage from trichomes. So that sticky substance you see is actually a trap for insects to basically get stuck, and then the plant will digest the insects for a nitrogen source. And a similar type of mechanism, but taking advantage of microbes found in nature, has been exploited by certain maize and sorghum land races, whereby they secrete aerial root mucilage, which can provide carbohydrates to nitrogen-fixing bacteria, and therefore there's an exchange between carbon and nitrogen with the plant and the microbes. So this could be important for reducing the need for synthetic fertilizers. All right, so let me tell you kind of what brought me to synthetic biology, and that is, you know, on the tail end of some very successful genetic screens on plants. So during my master's studies in Canada, I was looking for cell wall mutants, and we ended up discovering and characterizing this flying saucer 1 mutant, which turned out to regulate mucilage properties. But to my surprise, this was not a gene affecting carbohydrate-related enzymes. It was actually a ubiquitin ligase, so something that is involved in regulating the actual catalysts. So that was a little bit surprising. So for my PhD, once I moved to Germany, I decided to really hunt down those carbohydrate active enzymes. And I performed a reverse genetic screen based on, for mucilage-related carbohydrate active enzymes, using some of the known genes as the bait and bioinformatic tools, mostly leveraging transcriptomic data to kind of predict which other glycosyltransferases or related enzymes might be involved. And relative to our wild type shown here, this is the Columbia 0 seeds for Arabidopsis, I found a series of mutants. Here I'm showing you just 2 of them, which led to noticeably different mucilage properties when mutated. And these actually led to the identification of polysaccharide synthases involved in the elongation or decoration of several classes of polymers. And through a research exchange to INRA Research Institute in France, I was also able to discover that some of these cell wall mutants actually occur in nature. And these are different ecotypes of Arabidopsis collected from different regions around the globe. And so there's the potential that certain cell wall compositions, we may view them as defects, they could in fact provide local adaptation or benefits in certain environmental conditions. So that's all well and good. We have identified some of the biosynthetic players. Some of them had very obvious defects. For example, most of the mucilage in our Arabidopsis seeds is composed of pectin. Those are gelatinous polymers. You'll find them in jams. There's also cellulose microfibrils. Those are the most abundant fibers in nature. But there are also hemicelluloses, such as galactoglucomannan and xylans. These are involved in cross-linking the cellulose microfibrils and forming connections to the pectins. Now, some of the mutants had very obvious phenotypes, like shown in these images. But more recently, we've identified there are classes of mutants which were anticipated based on the work of other researchers, like Nick Carpita, a pioneer in cell wall biology, that actually have invisible phenotypes. That means a mutant may look like the wild type, but in fact, in terms of biochemical composition, those lines could have significant changes. And those could be promising targets for synthetic biology because it means you may be able to fine-tune the composition of a particular cell wall without leading to growth defects or some other penalty. So I'm going to focus the rest of my presentation in terms of using cell walls as a case study, focusing primarily on a single class of polysaccharides, which are the beta-mannans. Beta-mannans are hemicelluloses that technically have the simplest structure possible. They could be composed exclusively of beta-1,4 linked mannose units, as shown up top, or they can also include beta-1,4 linked glucose bonds, and they are structurally analogous to cellulose. If they don't have any side chains, they can form hydrogen bonds and have really crystalline properties. But they can be substituted. That's what distinguishes hemicelluloses from the crystalline cellulose microfibrils, and that leads to enhanced gelling properties. Mannans as a whole are involved in carbohydrate storage. They can be akin to starch in cereals, but they are most abundant in legumes. They're not exclusive to legumes. For example, here I'm showing you konjac noodles. These are produced from the corm or tuber of the Amorphalus konjac plant. And such polymers also accumulate as storage materials in non-legumes such as coffee seeds. And that's one example worth pointing out because coffee originated in Ethiopia. So for the purpose of this audience, I'm not sure how many of you are aware of your mannan consumption, but it's very likely that you're ingesting some of these polymers on a daily basis. They're also added as stabilizers and gelling agents, for example, under the names of guar gum or locust bean gum, to numerous foods in the supermarket. If they were unbranched, they actually form an excellent carving material. Here are some palm seeds used as a source of vegetable ivory, so no animal has to be harmed. And in the case of softwood, that is the wood isolated from evergreen species, gymnosperms. They are the dominant hemicellulose. So it is really important for the paper and timber industry. And we think we know based on a few isolated examples, they're also important for plant biology. For example, a single mannan synthase mutant in Arabidopsis is embryo lethal. So that points to the important roles of these polymers. So why do we want to use synthetic biology? Well, we can learn by building these polymers, right? If you make a plant mutant, you see what's left, what happens as a consequence, but you may not be able to just re-engineer or increase the content of that polymer. People have tried, and there were some unexpected bottlenecks. So we proposed several years ago to instead use a surrogate host to understand the specificity and activity of different mannan-related enzymes. And the main players are cellulose synthase like A or CSLA enzymes. These are the actual enzymes that elongate the backbone. They have multiple transmembrane domains, for example, 5 domains for the konjac CSLA3, which we'll just call mannan synthase or MANS for this presentation. And we can, express a single enzyme or combination of enzymes in a surrogate host and understand what are their individual or joint functions. We can then also investigate the metabolic bottlenecks. Is mannan elongation limited by the precursor or by the enzyme itself or by the need for a cofactor? So compared to traditional approaches in the cell wall field where people would express recombinant enzymes in a host like a yeast or insect cell or mammalian cell, purify the enzymes and then feed radiolabeled substrates for in vitro assays, my lab is focused on actually engineering the host to accumulate the plant-based polymers. And then we analyze the carbohydrate as opposed to having to extract the proteins, which is more time consuming and not feasible necessarily if you have a protein with multiple transmembrane domains. And we focused on yeast as a promising system because early experiments indicated the power of yeast genetic tools, the speed of cultivation, and the relative yield of polysaccharides compared to the cost of the simple media and the precursors. What you'll see by the end of my presentation is that much of the work that we've been doing for the last decade, and this is not limited to my lab, has been an artisanal process. That means, each student from the undergraduate all the way to the postdoctoral level actually needs to learn manual techniques that can be only carried out generally in small batches and require individual skill. And that is a limitation for advancing our knowledge and the development of products based on carbohydrates. So let me start by showing you that using yeast as a model system, here we use the yeast Pichia pastoris, we've been able to express a single plant enzyme. This is the mannan synthase from the konjac corm. And even within 9 hours of methanol induction for this yeast, we're able to see significant expression of our desired enzyme if it's tagged at the C-terminal end, and that translates to the significant elevation of mannose-containing alkaline insoluble polymers. So this C-terminal tagged enzyme is active. If you wait more than 9 hours, like a day or two of induction of protein expression, you don't necessarily get more mannan. It was almost saturated from the start. And we could look at, because these are cells with fluorescent tags, we included a known Golgi marker, SCGOS1, and our tagged mannan synthase. And we could see that there's co-localization of the plant enzyme with a yeast Golgi marker. So in fact, our orthogonal system is producing the hemicelluloses inside the cell, excuse me, in the Golgi apparatus, and its products are likely secreted to the cell wall. All right, then we could ask some questions. Okay, what are the bottlenecks for the production of plant mannans? In yeast, we have a variety of mutant collections as well as work done by other groups. And one of the reasons we focused on the yeast Pichia pastoris is that we knew it had a lot of mannose precursors. The yeast normally uses it to glycosylate proteins. And so this is what in wild-type Pichia you can find. Surface proteins can be highly manosylated. Those are all the little green circles. But there is a commercial Pichia glycoengineered strain called Superman 5, which has reduced the manosylation units to only 5. So we hypothesized that this background would actually free up mannose, especially the GDP mannose, that is the activated precursor for mannan biosynthesis, to produce more plant hemicellulose. So we took our mannan synthase tagged with GFP, and we introduced it in Superman 5 strains, which are used industrially to make therapeutic proteins and other products. And surprisingly, the protein still expressed well, but we did not increase the amount of mannan made compared to the Superman 5 control or our wild-type Pichia controls. So that actually indicated that no, the activity of this enzyme does not appear to be limited by the amount of precursor. In fact, evidence I'm not showing you here indicates that there are other protein cofactors which come from plants that actually need to be present to make the enzyme work better. So we still had expression of our enzyme in both the wild-type Pichia strain and the Superman 5 and these intracellular punctate, consistent with the Golgi apparatus, but no elevation in hemicellulose production. So, this is a simple but elegant example of the power of yeast to answer questions that will be relevant for crop engineering. So moving towards crop engineering, okay, how can we decide what are we going to test in plants? I heard Renee mention in the previous lecture that plants are difficult to engineer. If you have to go through tissue culture, maybe it's a year before you've regenerated plants for analysis. So again, this is where yeast or other high throughput platforms can be critical to success. And we've recently adopted the plant modular cloning or MoClo standards, which were developed by Silvestro Mariane in Germany. He was a former colleague. Now, these systems, using a technique called Golden Gate cloning, essentially allow us to mix regulatory elements, coding sequences, akin to Lego bricks. You can decide how strong a promoter you want, which specific gene you want to express. Do you want a single gene or several? So our recent contribution has been to actually evaluate if we can use the plant MoClo standards to express plant enzymes in yeast, specifically cell wall-related enzymes. We could screen many enzymes in yeast in a matter of days and then decide to only prioritize some of them for more time-consuming experiments in planta. So in a bioRxiv preprint we released this year and recently revised, we started assessing whether off-the-shelf Pichia episomal vectors could be used to make plant hemicellulose in yeast. Episomal vectors are advantageous because they are very rapidly taken up by the cells. And compared to vectors that require genome integration, you will get much larger number of colonies. So you could theoretically screen for a large library of parts and be able to find out the winning combinations. Well, what we discovered was, here we have a GFP green fluorescence protein control, so no polysaccharides are made. This one grows fine. If we try to express the conjugate mannan synthase in this off-the-shelf Pichia vector with a fairly strong PCAT1 promoter, who we're not getting any cell growth. I mean, the strain is really unhappy. So that was puzzling because before we didn't have this issue. So what we did then, we started replacing the promoters. We took the episomal vectors and made a new version where we could introduce any promoter we want. And we reverted to the methanol-inducible AOX1 promoter, which is the gold standard for Pichia. And now you can appreciate here's an integrative vector that is commercially available. And this is the new L1 episomal vector compatible with the plant Moclo standards. So if we have AOX1 promoter, we are able to accumulate mannose, maybe not as high as the classical vector in this case, but we've avoided the growth penalty. So what we learned is basically that some promoters actually be too strong, and then if the cells are producing too much plant hemicellulose, they might not be happy. It can actually be a toxic product. So dialing in the right level of expression is quite important, even for surrogate hosts. And then we also showed that the same enzymes with Saccharomyces specific promoter, inducible promoter, could be used to produce hemicellulose in the common budding yeast. And this is used more broadly in the research community, as well as for fermentation of various therapeutic proteins, as well as beverages and other things. So this is quite a versatile system where we can now test a particular set of plant enzymes in one or more high throughput species. So the vision is this, that instead of spending minimum 50 days or even longer to engineer a plant, we could actually prototype our designs in yeast, where within 1 or 3 days you have sufficient polysaccharide production to detect what's happening. You could use that to improve your enzymes, even refactor the pathway. So for example, trying to address some of the bottlenecks, and then put the winning designs into the more time-consuming pipeline. All right, so moving on, we used such an approach to really probe the specificity of other cellulose synthase-like proteins. This cellulose synthase superfamily is found in all plant species with various copy numbers, and we knew which enzymes made cellulose, the ones that made mannans, the CSLAs, and a neighboring clade that made the backbone of xyloglucan, which is a common class of hemicelluloses found in most plants. So one of my PhD students in Germany, Annika Grisosowski, started wondering, well, what are the activities of other members of these families, especially the CSLK clade, which is only found in algae, and it's kind of proximal to both the glucan and mannan synthases, so it may be difficult to predict their activities. And so by sampling about a dozen enzymes from across the plant kingdom, we found that only 4 of the 9 CSLAs found in Arabidopsis actually produce mannan in our yeast system, which aligns with the genetic evidence. But if you look at so-called early land plants, like the mosses or the liverwort, Marchantia polymorpha, where you have fewer copy numbers, they're all active. Surprisingly, the green algae, for example, chlorophytes or caryophytes, were assumed to make mannan, produce mannan, but we didn't find any positive evidence to support that. Meanwhile, the more distant related red algae are appearing to produce bona fide mannan. So that suggests that even if the red algal enzymes are producing mannans, perhaps there's been some functional diversification in the green algal clades we analyzed. Now, of course, I have to stress this was only about a dozen enzymes, and so it cannot be representative of the whole diversity of species found in those clades of life. So to address this issue, we actually needed to develop some new probes to detect carbohydrates. Current stains are unspecific, or if you were to use monoclonal antibodies, which are available for certain mannan motifs, they're quite time-consuming to apply. We wanted a probe that would be suitable for live cell imaging, and so we exploited carbohydrate binding modules. These are non-catalytic protein domains found primarily in bacteria and fungi, which are designed to recognize specific cell wall glycans. And so we took some mannan CBMs, tagged them with a red fluorescent protein, or RFP. This is work led by another student, Madeline Robert, and programmed them to be secreted from our PICIA expression system. Now we can introduce this probe in different yeast strains. We have our wild-type yeast cell walls, mannan or glucomannan producing strains, and we looked at the supernatant or the media that remains after these cells are grown and centrifuged. And so what we found was that if we look at the fluorescence measurement of the supernatant, the wild-type yeast background has a high secretion of the CBM RFP probe, but whenever you make mannan or glucomannan, there's less signal in the supernatant. So we did confocal microscopy on these cells, and what we saw was that there's almost no visible mannan probe retained in the cells with the wild-type cell walls, but there is mannan binding probe retained inside the Golgi apparatus of cells that express an active mannan synthase. So this is providing a nice indication of whether cells produce mannan or not. We can make this into a more quantitative probe by adding a ratiometric aspect. So here we have a self-cleaving peptide that enables cytosolic blue fluorescent protein to be cleaved during the or spliced during the translation process and give us a kind of like a counter stain, so we know whether the cells actually have the probe. And again, we have our wild-type cell walls up here with no carbohydrate probe, our new mannan tract probe in cells with a wild-type background. The only thing that remains visible inside the cell is this self-cleaving cytosolic blue fluorescent protein. But if those cells actually produce mannan by expressing a mannan synthase, we have the mannan synthase, we have the counter stain, the blue fluorescent protein, and we have our probe being retained inside the cell. So now we really have a tool that can enable us to do high throughput screening of whether mannan is made, and also, for instance, take advantage of modern techniques such as cell sorting based on fluorescence to enrich for certain variants. And we've been able to put the same probes using plant promoters for constitutive expression in tobacco and Arabidopsis. And we have preliminary data that these can also be used to track the dynamics and distribution of cell wall components in a living plant without dissection or doing more time-consuming analyses. So here's some evidence that from a pilot screen of randomly mutagenized mannan synthases, that the mannan tract probe can help us screen for which variants are still active. So we have this randomly mutagenized enzyme collection, and we just sequenced a few varieties to see whether they have amino acid changes. And there's a fairly wide distribution of what the changes are. So for example, our first variant has 8 amino acid changes. As you'll see, we discovered that this still being tagged with GFP, we still had enzyme expression. That's good. Others had, for example, an early stop codon. Therefore, the C-terminal GFP is no longer made, and so there's no expression. And we could also see whether they produce mannan based on biochemical measurements. But if we look simply at this graph, at the CBM probe secretion, that's the mannan, fluorescently tagged mannan probe. Again, in the wild-type background, we have high secretion. If you have the parental version, very low secretion because mannan is made, so the probe gets retained. And then we're quickly able to see whether our new variants are functional. And And we can also correlate that with the mannan content. And so I'll just point out one example. So we have here variant number 9. It has very low mannan content. It has high RFP secretion despite having a GFP signal. So really, there would be no other way without destructive experiments, grinding the cells, hydrolyzing the polymers, spending a couple of days preparing the samples to know that this variety is not going to be functional. But with the probe, you can accurately predict that upfront. Meanwhile, the neighboring line, variant number 8, has low secretion, high mannan production, so we could prioritize that for further experiments. Okay, so we have a screen, a rapid prototyping tool. Now we really want to fine-tune the composition of the plant fibers for different applications. That was the goal I introduced up front. Now, thus far, we've talked about the CSLAs or the mannan synthases. They make the backbone, and that's sufficient to get some fibers made in yeast. But if you want to make something that is useful for, let's say, for new gelling agents or other properties, you need additional enzymes. In fact, you need enzymes that make the precursors, that other enzymes that add things in the Golgi apparatus, and then remodel the polymers once they're secreted. So in collaboration with the Joint Genome Institute in California, I've had the opportunity to synthesize 500 kilobases of DNA, which will be used to probe the functions of different members of this pathway. Why this is important is that it enables us to make a large number of combinations to produce new to nature polymers. Now, the number you see there, 10 to the power of 7, is staggering and actually not feasible based on our current throughput for making the constructs, transforming the cells, and all that. In fact, even just analyzing 130 cellulose synthase like A enzymes could be a lot of work for a single student. Excuse me. So the Joint Genome Institute, or JGI, is sending us the coding sequences for the enzymes we're interested in. So how are we going to manage to analyze their functions? Well, in the first phase, we're going to get over 100, and we've received actually over 100 cellulose synthase like A enzymes. We want to prototype them at yeast, prioritize the best ones for later experiments in planta, as I outlined, and really take advantage of our mannan track probe to quickly identify which ones are still active. Another element I didn't show you is that the mannan track probe could be used both in liquid cultures, where you can grow cells in multi-well plates. It could even be used on solid media, where, for instance, you have fairly good growth of all the strains, but if you shine the right excitation light and have appropriate emission filter, you can detect based on red fluorescence which colonies are most likely to have active mannan synthases. The big problem to all of this, and it's nicely summarized by this neat diagram, is that we have the plasmids, we've done the design, everything's good. Now it's time to make our final constructs, putting the promoter, our expression vector. You can do all that and not learn anything if an error occurs. Why is that? I mean, any step of the pathway, when you incubate the cells with the DNA, when you add the antibiotic, when you do the assembly, when you do a PCR, things do go wrong. And often you go back to square one without knowing precisely which step was affected. So this is really where automation comes in and can help us make semi-automated procedures. In my lab, we've established an entry-level biofoundry we call the Botany Foundry, employing Opentrons robots and user customizable scripts. Now, why would we want to do this? We want to have affordable and accessible protocols, which can be scaled from low to medium to high throughput experiments, and they will help us minimize repetitive techniques, fatigue, the risk of inexperienced users having a pipetting error, and maximize human brain power. Before I go on to show you our protocols, I want to introduce some principles that I teach in my courses at the University of Florida, why do we even want to do this? I mean, ideally, you have a well-organized workflow. Things are documented digitally nowadays, but reality may be quite different. There could be messes, notes that get forgotten or never typed up. So I will share the slides after this class, but I encourage you to look at this short clip from Doug Densmore at the University of Boston, where he explains how restaurants, for instance, have highly automated procedures that could also serve as a model for biologists to kind of have reproducible workflows. So a biofoundry will have several phases, the key one being the design, right? Are you using an electronic lab book or some other software to design your constructs? How are you building those things? You now need to introduce living systems, organisms, cells. How are you managing the data so that you can do some tests and learn information that can help you iterate the design cycle? On the test side, it depends on what kind of molecules or process you're after. For carbohydrates, we use liquid chromatography as well as gas chromatography, mass spectrometry to analyze the biochemical composition of the cell wall. But as I mentioned, it's much more attractive if you can have a fluorescent probe or some other detection system. So there are facilities that can help you do this around the world. But as shown as in this map from 2019 publication about the Global Biofoundry Alliance, or GBA, there are some major areas of the globe that do not have access to this technology and automation. Africa being a notable omission, as well as South America. 4 years later, 2023, here's an updated map. The situation is not much better. I mean, even the southeast of the United States, where I am located, is sort of a zone that people in academia do not have access to automation that is concentrated in a few other parts of the U.S. and Europe and Eastern Asia. So some of you may have heard this quote, but it seems like the future is already here, but it's not evenly distributed. So this is a real challenge because you can have an incredible 6-7 figure facility available in Boston, but if that only serves a small community, then many are still held back by the manual procedures that I described. So there's different levels of automation you could start with. There's software automation. There's a nice video. I won't have time to show you now, but basically, instead of using manual timers in the lab, you could have a workflow that you can hand off to another student, and therefore they could more easily track what step they need to do next and how much time to wait for a certain incubation. The system that we've adopted are low-cost liquid handling robots from a company called Opentrons. When we started, they only had the OT2 system. Shortly afterwards, in the last year or so, they released a higher throughput Opentrons Flex robot. Even if they're not cheap, these robots are, at least an order of magnitude more affordable than some other commercial solutions. And they've also partnered recently with other teams that actually do research, like the folks at Neochromosome that are involved in making fully synthetic yeast strains where they can write genomes and tailor these organisms for specific applications. And even though the cost of these robots is increasing with inflation, they are still kind of the logical entry point for many research teams. Now, this is not an easy process, so I will, before I show you the methods, I just want to present the caveat that if you have not heard of SWOT analysis, there's strengths, weaknesses, opportunities, and threats for any endeavor. You know, the strengths should be evident, right? You can have standardization, automation, kind of tackle problems at larger scales, develop new products faster than before, but you're not going to have a system, a set of robots that can do everything, right? It can become obsolete. In fact, if you buy $1 million system that cannot be upgraded or maintained or used by students. And, you know, there's a number of other opportunities and threats. For example, you know, in the context of plants, the regulation of genetically modified organisms is still a tricky proposition in many parts of the world. So let me, in the final few minutes, let me show you what our workflow is and how it's speeding up our research. And this is work that has not been published, but is very close to public release. And so we're happy to assist anybody else who's interested in employing these techniques for their own research. So it was led by Moni Chiandi, a postdoc in my lab, together with 2 undergraduate students, Leanna and Abigail. And we can cover everything from, you know, DNA extraction to preparing PCR reactions, DNA assembly following the plant MOCLO standards, which are also compatible with yeast, as I mentioned earlier, to transforming microbes such as E. coli, and then extracting the newly assembled plasmids in higher throughput than before. The robots are modular, so you can have any configuration you want. I will caution that it takes time to switch the configuration, and so that's why we have several robots now, each generally tailored to a specific kind of reaction. So our procedure generally starts with DNA extraction, and we make our own buffers. Undergraduate students can do this. We buy generic silica spin columns. So I think this would be an attractive solution also for any other research team that is conscious of the costs of certain brand name reagents or kits, or they do not have reliable access to these, some of the big commercial solutions. We can use centrifuges or vacuums to aspirate the parts, the wash solutions that contain contaminants. And then we generally get from 10 to 15 micrograms of DNA per spin column with fairly good purity. So now we want to, you know, we have DNA, we want to assemble a certain product. That is fairly simple to do using the robots. You just need a 1 channel pipette, for example, with a 20 microliter tips. And then we can load the components. For example, we have a temperature control module here with a master mix containing enzymes, ATP as an energy donor source. We can then load the DNA parts we want to mix together to have promoters, coding sequence terminator, acceptor vectors, and then the PCR fragments. And then instead of having to learn how to write scripts using Python, which is the programming language for Opentrons robots, we have developed simple tables where students just have to say, okay, I want the master mix from source well A1, that was temperature control block, to go to the strip tubes over here, small volume reactions, 5 microliters of each. And we can even say, like, pick up a new tip every time if you're doing, let's say, different DNA amounts, or just reuse the same tip. All right, so we specify everything in a convenient format. You can do, you know, kind of unique plasmids one at a time, or you can multiplex reactions, which means, let's say you have 5 different genes. You want to find out which of those 5 genes works best. Well, you can throw them all in the same tube by having, you know, a single destination for these different coding sequences. So now they're all going to the same destination PCR tube here in A1 of our PCR plate. You could also run 96 different plasmid assemblies at the same time. That's kind of the maximum possible with the current capacity, since we have 11 flexible slots on this Opentrons 2 rack. So you can have 96 different genes, 1 master mix containing your destination vector, and they can all be prepared in this final plate. Thanks to a HEPA filter that can be mounted on top, and we have to say this is a fairly expensive addition. It's costs as much as the robot itself. We can then have aseptic airflow underneath the, in the working surface. Can also use a thermocycler module to have temperature conditions that can be rapidly changed. And this allows us to do bacterial transformation. We can load a single tube of competent cells, some assembled DNA, some media for outgrowth, and automate this procedure where we take 20 microliters of competent cells, mix them on our thermocycler module with 2 microliters of the DNA, heat shock the cells, add the recovery medium, incubate at 37 Celsius for an hour, even without shaking, and then finally plate the transformed cells. Once you have colonies growing on a plate, we've written a protocol to extract DNA in high throughput using magnetic beads. And this uses a commercial kit from Zymo, where there was a published protocol available, but we realized it had to be completely rewritten. So there's kind of multiple steps to this procedure, and some of it we realized have to be done manually because we have to use a particular magnet stand, and the pipetting is trickier for the robot to achieve. But at least half of the workflow is done using the robots, where the DNA can be washed and finally eluted using a magnetic module where the magnets can move up and down inside the robot. We're also using 8-channel pipettes for this, so it is fairly high throughput compared to the manual DNA extraction if you had to do 96 samples in parallel. Now, the DNA has lower yields than our conventional silica column-based approach, but it is still suitable for transformation, not only of bacteria, but also of yeast. And so here we took some episomal plasmids, we transformed the yeast cells, and we obtained around 100 colonies for several different plasmids. And generally, it's sufficient to screen 3 to 6 colonies for a particular construct. So this is more than enough. We could even use it to screen multiplex constructs. And we genotype some of these resulting colonies, and they all contain the desired plasmids. So that's great. So finally, I gave you a few examples of procedures that can be done, but really the protocols that we're going to publish require no coding skills. They contain tables that allow you to customize, for example, which tip you want to use, what labware, what initial liquid volumes you have, what liquids you want to transfer and to where, with drop-down selection menus to avoid error. And you just load this CSV or comma-separated value table into the Opentrons software and be able to run it. That is much simpler than what's currently available. And so here's a final example from our research. We have a 96-well deep plate where we decided to inoculate yeast cultures in 500 microliters of media. Now, each color indicates a different yeast strain. They are colored, red, green fluorescent proteins, and some have controls. It would be quite hard to do this manually, right? You'd have to pay attention that you're inoculating the appropriate well, but the robot can do this without much difficulty. And if we look at the fluorescent plate reader measurements after growth, what we see is, for instance, green fluorescent protein is detected only in those randomized cells that were inoculated with the GFP strain, and the same for the red fluorescent proteins. So, if you had, for example, just water, there's no cross-contamination. It's everything worked out beautifully. So recently, this spring, I started teaching some of these principles to undergraduate students at the University of Florida. And this is a work that was done in partnership with Opentrons Education Initiative. So we actually have a course that you can download online from the Opentrons website, where again, no coding skills are required, and yet students actually go from not knowing how to pipette to cloning their own carbohydrate active enzymes within 12 classes. Instead of showing you all the cloning verification steps we did, I want to show you a nice collaboration done together with another tool for Opentrons called BioArtBot, where essentially we printed fluorescent or colorimetric bacterial cells. And these are student designs that they just inputted into a graphical interface, and then the robot did all the work. We incubated the cells overnight, and this is the final outcome. The cells, in some cases, have fluorescent proteins. And so here's an image of some plates under white light in a gel documentation station. But if you apply the right fluorescence filters and settings, you can actually see the fluorescent signal stand out. And so you can imagine, thinking back to our fluorescent probes for carbohydrates, we could actually develop a high throughput screening platform where we can pick from a solid plate which colonies are worth regrowing to produce a particular polymer. So I hope I've demonstrated the power of synthetic biology to characterize plant enzymes, especially challenging ones like glycosyltransferases in yeast. How we've developed fluorescent probes to detect specific carbohydrates. And the fact that now we have access not only to automation that can accelerate discoveries, but would be accessible to other researchers around the world. And we think there's great opportunities to incorporate more plant-specific procedures, such as agrobacterium-mediated transformation using Opentrons. And part of this has been demonstrated by the Hasselhoff lab from the University of Cambridge earlier this year, and also by high-end biofoundry at the University of Illinois, which I have to point out, I mean, they have a high degree of automation. So if we could take some of their procedures, like preparing protoplasts, and achieve the same result using a much more affordable platform, again, we can democratize the technology. So with that, I want to thank the people from my lab that did the work, some of the funding sources, previously in Germany, but especially since my move to Florida 3 years ago, and you for your attention. And I realized this was a little bit longer than I anticipated up front, but I'm happy to take any questions and stay for an extra 15, 20 minutes if need be.
Thanks a lot, Catalin. That was very great. I really enjoy it. I feel like, a lot of the Opentrons work can help, with the teaching in the further area for sure. There are also other things that we have been in touch, like if we're to explore something that we do the teaching in Africa, in which like it would be not that easy to ship everyone's who can teach to Africa. Like a lot of remote teaching, including this kind of methodology, is very useful. So for the audience, if you have any question, please feel free to type or just like raise your hand, and we can like have maybe like 10 or 15 minutes discussion when Catalin is still available. Maybe I'll start with my question very quickly. So for the Golden Gate assembly to make the plant engineering plasmids, I believe like we have to pick some colonies and do the midi prep, like you show. Is there a bypass step in Opentrons that we can do the colony picking? Because that was kind of, I might have missed it, but like it was like one thing that was kind of a very expensive apparatus so far that I'm aware. So I wonder if there's any like improvement on that.
So there's a great point. Colony picking would be a advantageous step. I mean, if you could do this using robots that don't cost 6 figures,
Yeah.
again, it would democratize the technology. There is a team, an iGEM team that set out to do this. I believe they were based in Germany. So Opentrons, the big advantage compared to some of the traditional players like Hamilton when it comes to robotics, is that they are open source, Python-based programming. So technically, if you have friends who are engineers who can manufacture or take off-the-shelf components integrated with Python-based programming, you would be able to introduce new functions. Cameras can be installed. In fact, there is a little camera already in the Opentrons, but it is not really functional. It doesn't have the resolution to detect, say, tiny colonies on a plate. So I know, at least of one team who tried to do this, so there's some potential there, but at the same time, what we learned with the DNA extraction, the 96-well magnetic bead assay, is that sometimes, yes, there's a, version 1.0, there's been an attempt or a protocol shared. Unfortunately, it may require, like in our case, a complete rewrite to actually get it to work. So, there's a benefit to this community that things can be freely exchanged. Opentrons has a free library of protocols on their website. So I encourage everybody to look for that. I'm not sure that the colony picking will be easy to implement. But the DNA extraction, for instance, now we finally have a working method. So that's part of the reason we want to share that with the community. It was not easy, put it this way, to get it to work.
That makes sense. Yeah, like so far when I've seen like any colony picking machine, it just like seems very hard to access to. And it takes a lot of training. I think it was like earlier in the days of the AI imaging where you need to train the machine sufficiently. Otherwise, they don't know which colony to pick,
Yep.
especially if we install other like, like the color or the fluorescent into the colony that we want to pick. Yeah. So, like there's some question on the chat. If some of you want to raise your hand and just like speak it out, like feel free to. I spot one question about the open course material. I think I found the link from the Opentron, and I'm gonna like put it in the chat right away. So if it's wrong, Catalin, please please help me like correct it. But I think that's the
Yeah,
one.
I think you got it. Yeah.
Yeah. So, Kato, take it away. You have a question.
Okay, thank you so much, Catalin, for such a wonderful presentation. It is more beneficial, me as an individual who is speaking on a perspective of both the African side and also having shared some knowledge from maybe Europe. I see that it's great knowledge and great power, more especially around robotics. But if I try to relate with the side of Africa, you find like this kind of technology has not yet reached in Africa, in most parts of Africa, maybe in a few selected countries, maybe like Egypt or South Africa. But the rest of the African countries may not be having the knowledge. But some of the young generation, they have the enthusiasm, they have the zeal to learn and maybe get 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 the knowledge and see how they can apply it directly in Africa. So are there, apart from maybe the the available open source information online, are there some other opportunities into which African students can tap in and get to learn on different kind of knowledge, maybe like through collaborations, through maybe exchanges, or maybe through more open source information, but which is like maybe deep information, not just the surface information, like the deep information. Yeah, that would be good to also try and highlight about that issue for the young researcher, for the young student who is on this webinar. Thank you.
Yeah, I mean, that's a big question. So if I understood correctly, one would be, are the robots accessible to African students? I think that is still a serious concern. I mean, even here at the University of Florida, we are the flagship university in the state. It's the first time that our undergraduate students had the chance to see and program a robot. I mean, it is not common, even in the United States, especially for biologists. I mean, if you're an engineer, right, you may have access to some manufacturing facility or something where you build your own robots. For biologists, this is still brand new. So I think I can make two suggestions. One is I meant to say up front that I'm going to share the PDF of my slides. So some of you can follow the links and some of the references I suggested, and feel free to reach to me via email if you have a specific follow-up question or resource. What I think might be possible, but don't quote me on this, is that Opentrons has made efforts to actually provide robots to the community for educational purposes. So naturally, they probably started in the United States where they're based. I know teams in Europe have won robots. I'm not obviously the iGEM competition that's been mentioned a few times. It's expensive to participate, but I wonder, if somebody has the initiative and the desire to say, like, I'm in a city where students want to learn how to program, they're interested in biology. Could you help us? Loan us a robot or give us a system that we can use to educate others. It would not be a big ask. I mean, it's part of the company's mission to actually educate users and train the next generation. So I would encourage people to follow that, and you can use for example, the course that I published as a reason to say, look, we're interested in plants. There is a protocol available, but we need the hardware, right? This is not an insignificant expense, but one that's worth trying to get company support for.
Yeah, I think that's a very great
Yeah.
point, Catalin. I want to echo another thing that I'm volunteer with. There was a class at MIT from the Community Biology Initiative, and the course was called How to Grow Almost Anything. It was
Right.
open for everyone. So part of How to Grow mission in the past year or two is trying to expand the nodes, and they were successful at getting Opentron support to ship the Opentron machine to different countries in the world. So we consider them a node, and a lot of the students were able, sorry for the noise. A lot of countries were able to like go to the community and like try a few experiments. Again, is a great work from Opentrons that help us, acquire robot for some initial work. There are still a lot more things to prepare, right? Because like the Openton doesn't come with infinite amount of pipettes, and we still have to like install several things. So like Openton has been very grateful and like supporting to the community, but we also need other supports from the other communities to make this work. And like it was like MIT was privileged to get a lot of support from the other companies to help like fund the class, and that's why like the class has been going and has a lot of supports going on. So those of you who might be interested, just like reach out to us or like check HTGAA website. Just like type HTGAA How to Grow Almost Anything, you'll find that as well. I'm part of the core TA. And that would be something that you can see whether that could be done at your space or not. I may have to say that like the course just finished in the past few months. That is opened up every year. So there was a plan to collaborate with the African region as well. I see Cornelius raise a hand. So Cornelius, please, if you have any question, please feel free to say it out loud. There are a few questions in the chat. I'll take a look.
Maybe before Cornelius coming, I think the issue of Cornelius, I have answered it in chat. He first asked it in chat. If it's the same, I have responded to it. Please, Cornelius, reach out to us via email because the question you're trying to raise is not related to this webinar. So please, there are some things we may not discuss on a webinar if they're not related with the webinar. Just send us an email. We can always organize a call with you. I personally can always organize a Zoom call with you and discuss with you whatever you want to discuss. Maybe there is another question from Katie. He says, I don't know if it's he or she. Nice presentation. My question is on Moklo, I hope. The automation itself is interesting. And he goes ahead and he's asking the kind of challenges during parts assembly for yeast vectors.
Yeah, so in general, we're going through the large scale assembly right now. What I can share is that the success rate has been very high. Typically for projects in my lab, because of the more time-consuming analysis we do downstream of obtaining a yeast strain or a plant, transgenic plant, carbohydrates take time to characterize, we tend to work with maybe 5 to 8 genes per person at a time. Now, that could be done manually, or it can be done with a robot for the sake of having more reproducible workflow. Just recently with the Joint Genome Institute constructs, we started working with like 80, 90 plasmids in parallel. Now, that is not something you want to do by hand. So the robots have been very handy. And we did, for example, 25 separate assemblies. That was our pilot screen. 25 separate assemblies. All of them were correct. We then tried multiplexing, so 5 genes in a single plasmid, but having several pools of these plasmids. That worked as well. So we think that there's no, so far the protocols are ready to deploy. When we have encountered an issue, generally you then have to diagnose which of your parts, for example, maybe the promoter was not correct, or you grab you grab the wrong tube from the freezer. When you loaded the robot, you may have made a mistake. But by and large, even for PCR, 20 PCRs, like 80, 90% of them worked when it's trying to amplify a new gene using the robot. So it's as good, if not better, than manual pipetting, where you're more likely to get distracted and do something.
Okay, thank you so much. I hope it answers. Cornelius, do you still have a question which is related to what I have responded to, or maybe you have a different question for Professor?
Yes, Prof, I actually want to say this. Looking at our African scope, in regards to DNA extractions or molecular study, it is quite theoretical, and mostly we don't have the resource in going into details of the genomic makeup of organisms such as plants in extracting their DNA. It becomes so difficult. So most of the times we go to school and we learn the theory aspect of those genetic makeups of organisms. So we want to, I want to actually emphasize, if we can, in a way, have a practical hand-on on DNA extractions and other genomic practical study. It will also help, rather than this theory that we have been learning, continual learning. I think it is not helping us as much, though we have learned so much, but how to now practice it becomes difficult. So if you can look into that, it will help us. Thank you so much.
Great point. Great point, Cornelius. So I can say from my own training, and I was privileged to, I'm born in Eastern Europe, but I grew up, my family moved to Canada. So I was privileged to kind of have the chance to take courses on plant science that, halfway through my undergraduate degree, I went from, first I thought I was going to be an engineer, then doing something in chemistry, and then I took a plant science course with the lab component. And by working with the plants, that's when I discovered, okay, this is something really exciting and something I'll volunteer to do, for the summer. So naturally, there could be, regional barriers or challenges to getting those opportunities. But I would encourage all of you who think, maybe plant sciences are for you, or you want to get into synthetic biology or molecular biology, like find a way, if possible, where you can get that practical experience. It totally supersedes what you can learn in a classroom. I mean, you have access to so many recordings on YouTube and things that, how to grow almost anything. I mean, I watch those videos. They're excellent. So there's a lot of theoretical knowledge, recordings, things you can learn from others. But if you have the chance, no matter how small, to get started hands-on, it can be a life-changing experience. And I hope, most of you find a way to, no matter how small, to get some experience, see if you like it. And then, it can go from there. It can lead to more opportunities. And in the US, you probably all, you may have seen the news, like it is difficult right now for, to bring international students. And so we also have to say that, okay, there are some, even in the US, there are some institutional barriers that we are all having to deal with at the moment.
Okay, thank you so much, Catalin, for that feedback. But there is something ICE will echo on beyond to answer Cornelius very well, but also on my side, as the founder of SynBio4ALL Africa, I would love to assure the African students who are here that the team at SynBio4ALL Africa, what we are working towards, it's not only keeping to the theoretical side or online side. We are working hard day and night. We spend sleepless nights trying to find a solution to that problem, because I personally, I grew up from Africa. I know what is taking place in Africa, and I wouldn't like to keep repeating the same errors maybe which have been happening. So we are working around the clock to ensure that we can find a mechanism and a sustainable mechanism to try and bring practicals to the African researchers, students, and everyone in Africa who has interest in synthetic biology. And I would like to promise you that with the support of different individuals, with the support of different institutions, within a shortest period of time, which I may not describe, we shall fulfill the wish which any African person would wish to see in Africa, the practical wish. Maybe ICE can also echo more, relating it with the Experiment Foundation and the current plant course we are trying to run.
Yeah, so we just like mentioned that throughout the class and throughout our webinars as well. So I'll just like maybe leave it at that. Kato mentioned a lot of those that are very interesting already. So for the interest of time, I would like to like maybe give time to me one or two questions, just so that like we can wrap this up. Since Diego has a hand raised, maybe Diego, you wanna ask a question?
Thank you, Ice. Thank you very much, Professor Kathleen. I was fascinated by the feasibility of producing enzymes in orthogonal systems, especially the time saving in the experimentation process. What do you think? What other benefits of using orthogonal systems like PICA and other organisms bring to plant synthetic biology in coming years?
Well, another approach I didn't have time to present is that we can actually use the yeast system to really evolve enzymes. So one of the things that's been achieved just in the past couple of years with the advent of genome editing is that you can have plants which contain targeted mutations that might not be regulated as genetically modified organisms in certain places like the US, potentially in Europe in the future. But all of those traits that are based on CRISPR-Cas9 systems generally, they're based on you turn off something. Now that's a knockout mutant or knockdown with the possibility to evolve enzymes, whether it's using artificial intelligence, rational design, or directed evolution approach where you start mutating and selecting for the best performing variants. We may actually have the possibility to discover what are the right mutations, amino acid changes to introduce in a particular plant enzyme, which then with the power of genome editing, you might be able to go back in the crop and then make that particular enzyme for certain traits, say nutrition, make that enzyme better. Now, you could not achieve that without a surrogate system like the yeast, where you can rapidly improve that enzyme and then use the latest tools for genome editing to make a pinpoint mutation. That is much faster than conventional breeding, where you make crosses and like screen thousands of plants to see which one performs best. That is just one example, but I think it's talking to people from the industry, they are all aware of the power of synthetic biology, automation, and trying to incorporate that into their future research pipeline.
Thank you, Professor.
Welcome.
Yeah, thanks a lot. I think we have went through most of the questions. Also the one in the chat. I mean, there are some general ones. So if the question is not directly about the webinar today, I mean, please feel free to reach out to us. And we will talk about the general things throughout the course and throughout the other webinars as well. So like for the interest of time, maybe I would like to thank Catalin again for your time and for giving us a very very great insights on not just the plants in bio, but also on the automation side of the plant synthetic biology as well. I think this would be like very inspiring for everyone who are here. Catalin will share the slide with us. We will take maybe like up to 24 hours to process the recording, and we also share with everyone. Again, just in case like there are some parts that you might be having more questions on. And with that, I think we just like wanna end it here just to be aware of the time. And thanks again, Catalin.
Maybe just one thing I would like to raise up. There is a lot of questions which have raised up in the chat that I want to do research in synthetic biology. I want to collaborate with institutions in the US. The first thing I would like to tell you is that last year we had a webinar which was about how can you do research, how can you do collaboration with the US institutions. And that webinar was recorded and it's on our YouTube channel. Please, you can look on it. But if you remain with some more questions, reach out to us via email and we shall be happy to help you. Thanks so much, Catalin. Back to you, Ice.
Yeah, I think that's it. Evelyn, you have anything you wanna say as a parting words?
Well, thank you to the organizers as well, and to all the people who tuned in. It's really great to kind of reach out to the global community, and I think the important move that you're doing is recording and sharing these videos. If people know about the resource, it will not just serve the live audience, but could be useful, a year or even 2 years later. So very glad to be part of this effort, and I'll be using some of the lectures to share with other prospective students as well.
Thanks a lot, Catalin. That's very glad to hear. So maybe with that, we'll stop the recording now, and maybe see you again in the next webinar, or maybe like for those who are taking a class, we'll see you at the class next week. Thanks a lot.
Well, thank you.
Thank you.
Thank you.